are actually in the membrane. Membranes provide a means to communicate. They are the border of the cell, so they provide a means to tell the inside of the cell what's going on the outside and to transmit that. We call that signaling. Now, very, very, very important, what membranes do is they allow cells to store temporarily energy and to convert energy from one to another type of units. The whole mechanism of ATP synthesis requires membranes. And photosynthesis and respiration, as we know it, would not be possible without membranes. Okay, so those are the different functions that we have to consider, and we'll consider each of those later in the class. Then, if membranes are made up of proteins and lipids, what is the function of these two? When we relate that to the main parts here, so we have a barrier and then we have the other functions like transport, enzymatic activity, communication, energy conversion. And we can simply say the lipids provide the barrier function. The lipids are the barriers. The proteins provide whatever action it is that's going on in that membrane. So if it's selective transport, that's provided by a protein. If it's an enzymatic activity, that's provided by a protein. If it's communication, that's provided by a protein. So we are going to predict, if we look at different membranes, and if the function of that membrane system is mainly to be a barrier, that we mainly see lipids in there. And that is what we see. Here's an EM picture. It's a thin section of what we call the myelin sheet. And what this actually is just a cell that's very thin and it's wrapped around a neuron. And its only function is to do insulation, isolation. So this myelin sheet, we see that if we look at the ratio of protein to lipid, the gram for gram, in this membrane system, we see that there is very little protein and 4 times more lipid in there. So there's a low protein over lipid ratio, because the function is to isolate. If we go to the mitochondrial inner membrane, which is very important for the generation of ATP in the process of respiration, there are a lot of protein complexes in there to give that action, and indeed we see the protein-lipid ratio there is 3.6, so more than 10 times higher than in this myelin membrane. If we go to the outer membrane of the mitochondrion, just for comparison, which is more of an isolating function and has some transport functions, but certainly isn't as active as the inner membrane, we see that the protein-lipid ratio is less than 2-fold that of the inner membrane. Yeah, so all in all, if we go to the plasma membrane of a cell that is not so super active in transport, like the liver, we see a roughly protein-lipid ratio of 1 to 1. But if we take the cell membrane, the plasma membrane of a cell in the small intestine, which is involved in very actively taking up substances from the digestive tract, we see a very high protein to lipid ratio. So what we see is very clearly the protein to lipid ratio gives us a strong indication of how active the membrane system is. Are you good? Okay. Now, so we have the lipids, mainly the barrier function, proteins, the activity. Let's look at the lipids and then at the proteins to see how they function. So why do lipids form membranes? Lipids form membranes because of their structure. This is a summarized structure of the most well-studied membrane-forming lipid. It's not the most abundant lipid in nature, but it is pretty abundant in our cells. Now, we find this lipid also a lot in the yellow, in the yolk of an egg, if you're interested. And in peanut butter. It's a very important component of peanut butter. Alright, so what is the structure? We have 2 fatty acids, attached to a glycerol. Here in orange, we have glycerol. And on glycerol there are 3 hydroxyl groups. To the hydroxyl groups we can esterify acids. So remember, an ester is a bond between an alcohol, a hydroxyl group, and an acid. So the fatty acid with its carboxyl group here is esterified to this hydroxyl group of glycerol, and that's a second fatty acid. Then we have a phosphate, so phosphate is an acid, esterified into the 3rd position here on my glycerol. And then in this case, there is an alcohol, and this special alcohol is called choline, which is esterified further to the phosphate. So the choline and the phosphate are very polar, very hydrophilic, and the fatty acids are very hydrophobic. In other words, this molecule has a hydrophobic side and a hydrophilic side. We call such a molecule amphipathic. Amphipathic means that it likes 2 sides. So the hydrophilic head likes to be in water, that's the hydrophilic head. The hydrophobic tails like to be in a hydrophobic, non-aqueous environment. Really what's going on, the term hydrophobic is a little bit incorrect, I have to say, because nobody is hydrophobic out there. Everybody likes to be in contact with water. It's just that water wants to hydrogen bond with itself or with other polar groups. So really, what's going on is that this fatty acid part of the molecule is excluded from the hydrogen bonding network. Yeah, so I really think the term hydrophobic is not really correct, it's not the other way around. That's just to be very precise. So these guys are welcomed in the dance, in the hydrophilic dance, in the hydrogen bonding network, whereas the hydrophobic tail is not welcome in water. Therefore it's excluded, therefore you get the spontaneous formation of a lipid bilayer if we take a lot of these molecules and throw them in water. Okay, and that's why people put this molecule, phosphatidylcholine, also known as lecithin, in peanut butter, because peanut butter, ground peanuts, have oils in there. You want those to be nicely emulsified with some of the water that's in there. That makes the peanut butter probably cheaper and more easy to put on your sandwich. And the hydrophobic tails cover the oil, and the hydrophilic heads are in contact with the water, so you get actually sort of a single layer of this molecule around your little oil droplets in your peanut butter, just so you know. Okay, now, this is a phospholipid, like we see here, because there's phosphorus in it, called a phospholipid. These are not the most abundant lipids out there in the world. The most abundant lipids are these guys, called the glycolipids, which have the same fatty acid tails, the same glycerol, but then instead of phosphate and some other alcohol, they have a single or double sugar there. So these glycolipids that are very abundant in the chloroplasts of plants and in the algae and cyanobacteria that live in the sea and that make half of the oxygen that we breathe, yeah, they are really the most abundant lipids in nature. But it's still the same principle. It's still a hydrophilic head and a hydrophobic set of tails. Okay, then we have another type of lipid called the sphingolipid. It's based not on a glycerol backbone, but something slightly different, but still there are fatty acids in there. And then we have steroids. Okay, so here we have those other structures. So here we have the general structure of our phospholipid that I just showed. With the example here, the choline head group. We can have other alcohols esterified here. Do we care? At this point, not too much. What makes a difference is which head group is attached here. It may add an extra charge, it may make the head group a little bit bigger or a little bit smaller, and there could be some consequences of that. We'll see that a little bit later. Remember, all of these guys are based on two acyl groups, diacyl, ester fights to glycerol. So they're based on diacyl glycerol. That's the skeleton. That's the major type of membrane-forming lipid. There's another design possible where we have, instead of glycerol and two fatty acids, we have this structure called sphingomyelin. And it's made up of this molecule sphingosine. Then you add a fatty acid, that's here shown as the small r, bound here to this nitrogen. So that's a bond that looks like a peptide bond. This peptide-like bond in here. So that's sphingosine and a fatty acid, that together is called ceramide. And if ceramide has then a phosphate group and a choline attached, and we call this sphingomyelin. This is also a membrane-forming lipid. It's found abundantly in the plasma membrane, especially of neurons and related cells. Now we can also have sugar groups attached instead of the phosphate and the choline, and then we speak of cerebrosides or gangliosides. You don't have to remember, but what we should remember is that instead of diacylglycerol as a backbone, you can have a backbone based on ceramide. And that's also a membrane-forming lipid. And then the third lipid that can be abundant is cholesterol, at least in us, in mammals, and related sterols function in other eukaryotes. So this is cholesterol. It has also a fatty acid, a short fatty acid-like tail. It has here these ring structures, and it has here a very small polar group. This is its head group, and to the right is the hydrophobic tail. So you should be able to distinguish something that's based on diacylglycerol, I think it's simple, from ceramide, I think it's simple, because you look for this peptide-like bond, from sterols, which have these ring structures. So I think it's easy. Now, how do these things then form membranes? We get to this type of model where we have here the polar head groups, the fatty acids, and here in green the cholesterol is in between. Now this is a static picture, and we should never forget that in reality the biological membrane is very, very dynamic. There are immense amounts of motion going on there. And to put it in skateboarding terms, I should say the amount of motion and dynamics in a membrane like this is massive. Skateboarding terms, massive motion. Okay, so how do we know this structure is correct? I've told you, well, freeze fractioning gets you this one membrane part on one side, the other on the other side. You see the proteins in there. The proteins can be visualized in the EM. You can also see here in this just thin section part, high magnification, you see here the two polar head group regions, which binds to the staining heavy metal that's used in this case. And in between here we see the fatty acid layers, which do not pick up that stain. So blow that up, you get this type of structure. So the head group, head group, head group, head group, and in between the fatty acid layer. And see that in another membrane as well. We have a minute or two. I told you the shape of the membrane is very dynamic in there. So what kinds of motion do we have? There's a figure in the book, and it shows us these three types of motion. So first of all, we have the rotation. So if I'm a membrane-forming lipid with a head and hydrophobic region, and you have this motion. It's extremely rapid, but it can be measured. So extremely rapid, like millions of times per second. Lateral motion is the second type of motion, such as diffusion. Lateral shift is called in this figure. Also pretty rapid. So rapid that every second or so a phospholipid would rotate or move around an entire cell. So fast also. And then finally, the phospholipids could flip over, going with their hydrophilic head through the bilayer and flip over, but that's very slow. And to get that at any appreciable rate, you actually need an enzyme to help it out. Now, why is this motion important? If we think about this fast rotation, then actually we get an average shape of this lipid. You probably, when you were young, have watched cartoons such as the Roadrunner and the Coyote. And the Roadrunner was always playing tricks on the Coyote, sometimes putting him in a roll of toilet paper or something like that, rolling it out, throwing it in, and then you see that Coyote spin around very fast and becomes a cloud. That image. Think about that is what's going on with phospholipids in the membrane. It's like this cloud of hydrophilic mass and hydrophobic mass. And you put several of those together, all rapidly rotating, and you get half of the bilayer. And it's this dynamic structure that gives the membrane the barrier function. And that's where I want to stop today.